So, hello everybody. I hope you can hear me. So, I see that my co-chairs turned their videos on. So, I would like to welcome you also on behalf of EACR to this first virtual conference, which has the topic DNA damage response in cancer. And so I would first like to thank you to join us for this new experience. I hope that we will be successful until you like it. And very specifically, I would like to welcome those people that had registered for the conference in Cambridge and that we had to cancel because of the COVID-19. So at first, I would like to introduce my co-chairs, Conchita Vance. She joined us also because the conference was organized in collaboration with Astro. And the other co-chair is Tim Humphrey. So welcome to the both of them. And the apologies from Jessica Downs. She cannot join us today because of other conflicting dates. So as I stated, this will be the first virtual conference, and we thought it would be very important to fill the time that we all have in the home office with great science and with discussion about science, and we are all keen on hearing new stuff. And therefore, we try to organize a virtual conference that covers the 4 main topics that we had picked for the actual conference. So we will have 4 talks given by Steve Jackson, Gaia Legube, Roger Greenberg, and Peter Glaser. And the talks will be 20 minutes each, and this will be followed by a 20 minutes questionnaire. Between so we will have 2 talks, and then we are happy to have a 30-minute industry symposium from 10x Genomics, one of the event sponsors. So thanks for all those speakers joining us for this virtual conference. So to stick in time, we have a 5 minutes break in between each of the talks, and you may also be interested how to then come to the questions and answer section. So how you can ask questions. You are asked to, and you can already write your questions through the Q&A box during the talk, and then the questions. So we will try to answer as much as possible questions after the talk, at the end of each talk. And to do so, if you have a microphone, we will ask you to read your question or to give your question verbally when you are asked to do so. So you will have then to turn your microphone on. You can also follow the questions that the other people asked in the chat, and then you in the Q&A box. And you can upvote questions that you find very interesting. So you have 2 ways kind of to shape the Q&A section. And I think that's all that I have to mention to how it will be going. And so since you would like to hear not me, but our speaker speaking, I would invite Steve Jackson to come up to the stage, so to turn his video on. He will give a talk on cellular responses on DNA damage, mechanistic insights and applications in cancer therapy. So I hope that all the technique and everything will work, and wish us a very successful event this afternoon.
Okay, I should be available.
You can start, it's all fine.
Okay, I can't see myself. Is the video on? Yes. And you can see me, yes, because I can't see
I can see you, and you will have to share your screen with the slides now.
Ah, okay. So
share, there's a button below where you can share screen.
Yeah. Continue. Okay.
So now.
Apologies for that. This is the first time I've given one of these types of talks, but I'm thinking that it probably won't be the last. I'd just like to start by thanking Lorena, Tim, Conchita, and Jessica for putting together this conference. It's not quite in the format that we originally thought about, but it's wonderful that they've been able to work together with the EACR to put this together. In these unusual times, I thought that rather than giving a very, very detailed talk about the very latest discoveries in my group, I thought I'd give a more broad introduction and also a rather upbeat introduction to this arena. So I think the committee and EACR have been very wise to choose to focus on cellular responses to DNA damage because as we're all aware, DNA damages and cellular responses to DNA damage are of huge relevance to cancer. We know that DNA damage yields mutations, which are crucial for carcinogenesis. We know that defects in DDR factors, you'll hear about some of them today, BRCA 1, 2, ATM, and others, predisposed to cancer. We know that radiotherapy and chemotherapies, which are still the mainstays of anti-cancer therapy, actually work by inflicting DNA damage. The success is via effects on cancer cells, and side effects, of course, are by effects on normal cells of the patient. We're increasingly seeing that DNA damage response biomarkers have diagnostic and prognostic potential in cancer. And we also know that the DDR doesn't work in isolation. It interfaces with many other aspects of cellular function. And there are many other cancer relevant processes that have interfaces with DDR, including immuno-oncology. We've also learned over the last decade or so that DDR furnishes many attractive drug targets. Now, there are many different forms of DNA damage, and there's a huge number of labs throughout the world focusing on different types of DNA repair and DNA damage signaling mechanisms. My lab has been primarily interested in DNA double-strand breaks, which are particularly toxic DNA lesions. Double-strand breaks can arise through physical rupture of the DNA or through agents such as ionizing radiation and certain chemotherapeutic agents. It's worth bearing in mind, however, particularly in relation to my talk today, that double-strand breaks can also arise when other lesions, for example, single-strand breaks shown here, are processed. This can occur in various circumstances, but it's particularly evident during DNA replication. Okay. Now, in relation to DNA double-strand break repair, our bodies, every organism on Earth, has developed various ways to deal with double-strand breaks. And there are two broad categories of double-strand break repair mechanisms called homologous recombination and non-homologous end joining. And it's very, very important for the cell to choose between these two pathways because they don't work in the same way, and they're actually better in different contexts. So one of these pathways is non-homologous end joining, a relatively simple system, although the details are quite complex, where a broken chromosome, the two DNA ends are brought together, cleaned up if necessary, and then pieced together with this complex of proteins shown here. The other pathway, major pathway of double-strand break repair, and it comes in various flavors, is homologous recombination. And the key feature of homologous recombination is that it requires the damaged chromosome to enter into synapsis with an undamaged partner chromosome. And this is invariably the sister chromatid. So importantly, this restricts functional homologous recombination, at least in cells like ours, largely to S and G2 phases of the cell cycle, whereas non-homologous end joining can operate throughout the cell cycle. Okay. Now, there are various proteins connected to these processes, but I just want to highlight the fact that BRCA1 and BRCA2 are connected to HR, as is the protein kinase ATM. Whereas in non-homologous end joining, we have a range of proteins, but I just want to flag these proteins at the bottom, XRCC4 and DNA ligase 4, because they'll pop up again in a few minutes in my talk. What I hope to convince you today is that the way cells choose between these DNA repair pathways is very, very important. And that if this goes wrong, it can have toxic implications for the cell and actually clinical implications. Now, the stories I'm going to share with you today started a number of years ago when I founded a company called Kudos Pharmaceuticals. And the goal behind Kudos was to develop DNA repair inhibitors. And as we set out in the original business plan, one of the major opportunities was to take advantage of the fact that cancer cells, in many cases, have lost one or other DNA repair or associated DNA damage response pathways. Now, if we think about that loss, that is a difference between the cancer cell and the normal cells of the patient, we might be able to think about this, for example, in the context of a genetic principle which has been around for almost 100 years, synthetic lethality. Now, in this scenario here, a cell or an organism can tolerate the loss of one gene or the other without losing viability. But if both of these genes are inactivated or changed at the same time, this leads to cell death or organism death. And so in the Kudos business plan, there was the idea that you might be able to translate this genetic principle into a pharmacological application, not by knocking out gene B, but by inhibiting the product, not by knocking out gene A, but by inhibiting the product of gene A in this scenario here. Of course, this scenario here was suggesting that there might be a way of selectively killing a cancer cell, which is very reliant on pathway A, whereas the normal cells of the body, for example, would have this alternative pathway, pathway B. Well, to cut a long story short, with my colleagues at Kudos, and there were a large number of them, we developed a number of compounds, some of which have actually made it all the way into clinical trials. And the one I'm really just going to focus on for the rest of my talk today is a compound called olaparib, also known with its trade name Lynparza. So Kudos developed this compound as an inhibitor of poly-ADP ribose polymerase, PARP. Now, what PARP does is it promotes DNA repair. It works in various contexts, but in particular, it plays key roles in promoting single-strand break repair and also latter steps of base excision repair. So what inhibitors such as olaparib and other PARP inhibitors do is to stop the poly-ADP ribose polymerase catalytic activity of PARPs. And what that does is two things. First of all, it slows down single-strand break repair. And secondly, it ends up with trapping PARP on DNA because PARP is not able to carry out these auto modifications with the poly-ADP ribose, which promote its dissociation. Now, interestingly, PARP inhibitors are not particularly toxic to most normal cells. However, if we think about it in the scenario of replication, we could see that there would be a potential for these inhibited PARP species or slowed down single-strand break repair situations, yielding double-strand breaks if they're encountered by a replication fault during S phase. Now, as I mentioned a few moments ago, we know that the resolution of double-strand breaks in those contexts are dependent on homologous recombination, and we know that BRCA1 and BRCA2 and a host of other factors are connected. So to cut a long story short, through a very productive collaboration with Alan Ashworth and colleagues in London at that time, we and a parallel study from Thomas Halliday, Nicola Curtin and colleagues found that PARP inhibitors such as olaparib and its precursors and other PARP inhibitors are very, very toxic to BRCA1 and BRCA2 deficient cells. As you can see from this curve here, the BRCA2 minus cells are exquisitely sensitive to PARP inhibition, whereas the plus or the plus minus cells are not particularly sensitive. Well, these data were, I guess it was a bit of a eureka moment for us because it suggested that PARP inhibitors would actually be selectively toxic to the cancers or the cancer cells or even pre-cancer cells in patients that have inherited one bad copy of BRCA1 or BRCA2. This is because the normal cells in these patients retain one allele and are therefore, at least in part, function for homologous recombination, whereas the cancers in these patients have invariably, through genetic or epigenetic mechanisms, lost the wild type copy, thereby becoming impaired in HR. Well, olaparib eventually, some years later, became a registered medicine. It's a precision medicine. Olaparib is the first approved DNA repair enzyme inhibitor drug, licensed and sold throughout the world. If you like, it's the world's first synthetic lethality medicine. It's the first cancer drug targeting inherited predisposition, and it was launched with a genetic diagnostic, and that's why I'm calling it a precision medicine. Although it was initially launched to treat ovarian cancer, it's now received approvals for breast and pancreatic cancer. And this conference is quite timely because just yesterday, the FDA approved Lynparza, olaparib, for metastatic prostate cancer patients with BRCA1, BRCA2, or ATM mutations. So as we're seeing with this PARP inhibitor, but a range of other PARP inhibitors that have been developed and commercialized by other companies, we're seeing that there is a growing potential for these as new treatment paradigms. And what we're also seeing is that the earlier these compounds are used in clinical management, the better the impact seems to be. Okay. But of course, this isn't really good enough. We know that even in these scenarios here, some patients are not responding, or if they do respond, they might be acquiring resistance. So we want to understand how resistance is taking place mechanistically, and maybe even think about applying that clinically. The approach that we've been carrying out the last few years is to turn things around and use a principle that we are calling synthetic viability, genetic suppression, where we're looking at situations where we treat a cancer cell, such as a BRCA2 deficient cancer cell being treated with a PARP inhibitor, under conditions you'd expect it to be killed, and then are doing genetic screens, in this case CRISPR-Cas9 genetic screens, to look for genes which, when changed or inactivated, will allow the cell to reroute DNA repair and survive. So we have a number of examples of this now in my group, but also many other groups throughout the world have done similar things. One of these was published a couple of years ago, which allowed us and actually other groups to identify a protein complex called shieldin. And this was through basically screening for BRCA1 mutant cells in the presence of various PARP inhibitors and doing genetic screens to identify genes which, when inactivated, would alleviate that sensitivity. We identified a number of proteins, genes in this kind of way, some of which were already known, such as 53BP1, you'll see there. But we also identified these, at that time, uncharacterized proteins, which we and others have shown are part of this shielding complex. And what we now know through mechanistic studies is that shieldin at 1, 2, together with another factor called shield3, work together with 53BP1 and other proteins to promote non-homologous end joining, at least in part by restricting resection. So a normal cell will able to carry out non-homologous end joining through this mechanism, but it can also carry out homologous recombination because a key function for BRCA1 is to counteract shielding, we believe, therefore promoting resection and allowing these breaks, when appropriate, to channel into the HR machinery. So cells without shielding are radiation sensitive because they can't carry out non-homologous end joining so effectively. BRCA1 mutant cells are hypersensitive to olaparib and other agents such as topoisomerase 1 poisons. We believe at least largely because this negative effect of shielding cannot be counteracted by the BRCA1 protein together with its collaborators. And of course, under these scenarios, if you don't have shielding there, then you no longer need BRCA1 to carry out a homologous recombination, at least to some degree. So these data show that one key function for BRCA1 seems to be to counteract shielding. Okay. We've explored the clinical potential of these kind of findings with colleagues here in the Cambridge Cancer Centre, and rather encouragingly, we are seeing that loss of various components of the 53BP1 shielding axis appear to be genetically or epigenetically downregulated in PDX models where resistance is either innate or acquired. Now, we also know that PARP inhibitors are being exploited in ATM deficient cancers, and this might make a lot of sense given the connections of ATM to homologous recombination. A couple of years ago, we decided to see whether or not similar resistance mechanisms would arise in ATM deficient settings, the similar mechanisms, for example, to those I've just described for BRCA1. But the answer turns out to be no. There is a resistance mechanism, but it's very different. So in this study, we started out, actually these were with mouse cells, either proficient in ATM or knocked out for ATM. And as we would expect, the ATM deficient cells in assays such as you see here are very sensitive compared to the wild type controls to PARP inhibitors such as olaparib, but also to other agents that generate one-ended double-strand breaks during S phase, such as topoisomerase inhibitors, such as camptothecin and topotecan. So the basis of this screen, carried out by these three individuals, was to take a concentration of topotecan that would kill the ATM deficient cells, but not the wild-type cells, but then do this screen on the ATM minus cells that have been transduced with a genome-wide CRISPR-Cas9 library, okay, to knock out all the individual genes one by one in different cells. Now, this screen turned out to be quite striking in its outputs. We identified a range of predicted drivers of resistance, but basically these comprise the XRCC4 and ligase 4 proteins, which are involved in non-homologous end joining, and also most components of a protein complex called the BRCA1A complex. Now, if you carry out a screen like this, of course, you need to validate the hits. And so, for example, I'm sharing you here with data that we generated focusing on ligase 4 and XRCC4, but we also validated the BRCA1A complex components also. So as you can see on the left here, ATM wild type versus knockout. The ATM knockouts are very sensitive to topotecan. If we take these ATM knockout cells and then mutate ligase 4 or XRCC4 with CRISPR-Cas9 genome engineering, we can see that this now gives resistance. And this resistance is quite dramatic. So if you look on the survival curve on the right here, the black line at the top is ATM plus, the red line is ATM minus, and almost in line with the top line are the double mutants. So this is a very strong suppression that we're seeing in these scenarios. We did the screen with topotecan, but as we expected, this resistance mechanism also applies to PARP inhibitors such as olaparib. So for some time, we were actually quite puzzled by this. We're seeing that loss of one DNA repair pathway, ligase 4 XRCC4 end joining, is somehow allowing ATM deficient cells to survive under conditions that they wouldn't normally do so. Well, a key experiment turned out to be this one here, carried out by Joseph Foreman. He point mutated the cells in the catalytic site with DNA ligase 4. So this is a point mutation. The protein's still making DNA ligase 4, but it's not catalytically competent. So these cells can do everything except the final step of non-homologous end joining. As you can see here, this point mutation also makes these cells much more resistant. So what we think is going on, and I haven't got time to provide the data, is that these types of breaks being generated during S phase should be repaired by homologous recombination, but in the absence of ATM, some, not all of them, some of them are being repaired by toxic non-homologous end joining. This toxic non-homologous end joining is, we think, leading to chromosomal aberrations and translocations, which then lead to mitotic catastrophe or apoptosis. So just to round this off then, in terms of a model, what we think in normal cells, ATM positive cells, is that they can carry out end joining when appropriate, they can carry out homologous recombination, but these double-strand breaks generated by things such as topoisomerase 1 inhibitors or PARP inhibitors need to be channeled into homologous recombination. And one key function for ATM is to channel these breaks into the appropriate pathway, in part by speeding up resection, and also secondly, we think, by counteracting non-homologous end joining. In the absence of ATM, we have delayed resection and lack of inhibition of end joining. And this allows some of these breaks not to be repaired by homologous recombination, but instead become repaired by toxic end joining, which is the large mechanism for killing these cells. And of course, if you don't have that toxic end joining pathway, you can now restore cell survival, the resistance to these agents, because there is no potential for them being repaired by this toxic pathway in this scenario. For aficionados, I just want to point out that we think we understand what the BRCA1A complex hits are doing. One of the key functions we believe, based on work from other labs, including that of Roger Greenberg and others, is that the BRCA1A complex is playing a role in controlling resection. And if we downregulate or lose components of this pathway, this enhances the speed of resection, probably therefore allowing the large majority of these breaks to escape from non-homologous end joining. So finally, I just want to round this off with just thinking about how this might have impacts in the clinical arena. Of course, understanding the mechanisms of resistance doesn't really help an individual patient whose cancer may, for example, have evolved resistance. However, I think there is a very exciting opportunity that our data and other data in the literature is now providing. So although resistance may have evolved for one drug, what we're seeing, for example, in this scenario here, in the context of ATM deficiency, losing XRCC4, ligase 4, or XLF is reducing sensitivity to camptothecin, topotecan, or olaparib, but actually is causing these double mutant cells to be even more sensitive to IR than the ATM mutant cells on their own. Similarly, in the absence of shielding, we're seeing that shielding loss is alleviating the sensitivity of BRCA1 deficient cells, but the double mutant cells are actually now much more sensitive to agents such as ionizing radiation and DNA cross-linking agents. So just to end up with an optimistic model for the future is that if we can understand ways of targeting cancer cells, and if we can then understand ways that these cancer cells may evolve resistance, in many cases, these changes in the cancer cell giving resistance will also be giving rise to some other change that could give rise to a collateral vulnerability, allowing another drug or combination of drugs to be used in these resistant cancers. I'll just leave you with a picture of my lab on lab retreat. Unfortunately, we had to cancel our lab retreat this year because of the lockdown. Hopefully that will change for subsequent years. I've also got a fantastic set of collaborators who contributed to the work I've talked about today. I'll mention that I do have various disclosures to make, none of which I think impacts on my positivity for the subject that I talked to you about today, and I'll be happy to take questions. I see that everybody's got mute. Everybody's muted as far as I can see.
There we are. Sorry about that. So thank you very much, Steve, for a really interesting talk. And congratulations on Lynparza now being approved for treating metastatic prostate cancer, which is another great advance for you and for the field, I think. So this is we have a 20-minute question and answer session now up until 1:45. So I want to encourage as many questions and scientific interactions as we can, especially given our circumstances. So just to remind you how you can ask Steve a question, you need to type your question into the Q&A box, and you can also use upvoting features if you see a question that you might have wanted to ask and it's already there, or if you see another question that you quite like, then you can tick that as well. And basically, we're going to start working our way down the list. So I want to invite you to write your question, and then I'm going to I'm going to call your name out, and when I do so, can you switch your microphone on and tell us where you're from and ask Steve your question. So our first question is from Anthony Chalmers. So Anthony, can you switch your microphone on?
Is that working?
Yep.
I'll stop sharing my screen now as well. Is that what, would that be?
That would be great. Yeah, I think then you can see at least the chair. Yes. Okay. Thanks. And you.
Thanks very much, Steve. Great talk. So you touched on this question right at the very end. I was going to ask you about the role of shielding in the radiosensitizing effects of PARP inhibitors, particularly in BRCA mutant, but perhaps in BRCA functional cells as well.
Yes, well, of course, shielding, given its connections to non-homologous end joining, you would expect it to have an impact on radiation sensitivity, and that's certainly something we are seeing. You know, this isn't a pure NHEJ factor such as ligase 4, XRCC4, but it certainly does seem to be promoting that pathway together with 53BP1 and associated factors. We are yet to actually fully characterize the full set of sensitivities or resistances that are imparted by shielding loss. And I think we also have to bear in mind the fact that it is potentially connected to other DNA repair pathways, and that's also an arena that I think we and I'm sure others are also exploring.
Thank you.
Thanks.
Thank you, Anthony. Next question from Mark Wade. Mark, can you switch your microphone on and ask Steve your question?
Hi, yes, can you hear me?
Yeah.
Yep.
Yes, Steve, thanks very much for that. Yeah, it's really interesting. Clearly, there's already sort of 4 PARP inhibitors in the clinic. My name's Mark Wade, I'm at Astex. I just wonder what your thoughts are about targeting other ribosylases or hydrolasers in that PARP pathway, or your thoughts on that in general as an approach.
Well, of course, this is now a huge arena, and actually that there are a large number of ADP ribosyl transferases and related enzymes that use NAD as a cofactor. And some of those are connected to DNA repair, but others are connected to a whole diversity of other cellular pathways. So I think this is highlighting the fact that if you want to have specific effects, you'll need to develop selective inhibitors against these. And I think these inhibitors, as they're coming along, are turning out to be great tools, not necessarily, well, in the end, maybe clinically, but also tools to actually get at the functions of these PARPs, many of which have been known about, but very enigmatic for many, many years. And all I can say is that, of course, there are companies and academic groups that have generated and are exploring clinical potential of these other PARP inhibitors, as well as ADP glycohydrolasers of various types as well. I think it's a very exciting arena, and it's nothing that I'm directly involved in right now, but I'm following with a great deal of interest. Thanks, Mark.
Great, thank you.
So the next question is from Joe Morris. So can you switch your microphone on, Joe, and tell us where you're from and ask Steve your question.
Hi, Joe. University of Birmingham, Joe Morris. Nice talk, and so nice to see you and everybody in the community. So my question is about whereabouts you think the ATM is involved. We know that CTIP loss can rescue sensitivity of, for example, BRCA1 deficient cells from using toxic end joining. So do you have a feel for where the ATM is involved in resection? Is it long range or short range? Which do you think?
That's a great question, Joe, and it's good to connect with you. I was supposed to be coming to Birmingham in the near future, wasn't I? But I guess that's not happening either. So certainly one of the well-characterized targets of ATM, but there are so many, is CTIP, and I think that would be having an impact on resection. But we also know from proteomic studies carried out by others that for example, the ATM is also hitting components of the BRCA1A complex, as well as components of the non-homologous end joining apparatus itself. So I think this is going to be a very complicated thing to unpick, unless you've got some of the insights that I don't have. I think at the moment, we're still just moving into that arena to see exactly how ATM is functioning in that regard. But I don't have any exciting new insights, unfortunately, to share with you at the moment.
Okay, thank you very much, Joe. We have a question from an anonymous attendee, so I can't really ask you to turn your microphone on, but I'll pass the message on to Steve, which is, do you see similar effects by inhibiting the ATR kinase, Steve, as well as the PARP? Because of course that can affect resection as well. Yeah,
and as I'm sure many in the audience will know, that ATM is related to a protein called ATR, and other members of that family include DNA-PK, CS, which is part of the non-homologous end joining apparatus, as well as mTOR, which is associated with other functions in the cell. Over the years, various companies, including Kudos, AstraZeneca, a whole range of other companies, including some relatively newly arising biotechs, have generated ATM inhibitors, which are showing interesting clinical potential, as well as ATR inhibitors. Now, initially, you might imagine that ATR inhibitors would not be a great therapeutic, because ATR is a pretty essential protein for just about every cell. If you knock out ATR or take it away, cells don't like it, and just about every cell, well, every cell line I know dies when you take ATR away. So though that might suggest it might not be a great therapeutic target, it turns out that the ATR inhibitors have been developed. They are tolerated reasonably well, at least in animal models, and are now in the clinic. And I think the arena here is to think about identifying therapeutic windows whereby the ATR inhibitor is selectively toxic to cancer cells, but not normal cells. And I think one interesting arena where that is playing out is in the context of ATM deficiency. ATM and ATR are partly overlapping in their functions, and what's been quite clearly established in the literature for a number of years are that ATM deficient cells are more sensitive to ATR inhibitors than are ATM proficient cells. So that is one arena where ATR inhibitors are moving forward now in the context of ATM impaired or ATM deficient cancers.
Okay, thank you very much, Steve. The next question from Sarah Simonetti. So Sarah, if you can switch your microphone on and tell us where you're from. Sarah, are you there? Sarah Simonetti. Perhaps I can ask her question for her. So this is quite a clinical question, Steve, and it is, are PARP inhibitors effective for bone metastasis from prostate cancer, sensitive to anti-androgens? So I don't know if you're, if that's something that you can discuss, or.
This is putting me on the spot because I'm not a clinician and I'm not involved in the clinical trials. All I can say is that I think there is a great deal of excitement with clinicians in the prostate cancer arena. It turns out that in sporadic prostate cancers, a significant proportion of them have deficiencies in genes such as BRCA1, BRCA2, and ATM. Some of these are genetic changes, other are epigenetic changes, and that in these circumstances, these prostate cancers do seem to be responding to PARP inhibitors. Now, from first principles, I would expect that also to apply in metastases of these cancers as well. So I think there is real potential there. I couldn't really comment because it's not my arena how the hormonal status of those cancers might impact on their sensitivity or resistance. Although I would imagine that if there is a DDR vulnerability deficiency in these cancers, then that will play out irrespective of the hormonal status.
Okay, thank you very much. The next question, which has got a bit more of an immunological flavor, comes from Gamze Gune Eskila. So Gamze, can you turn your microphone on and tell us where you're from and ask Steve your question? Gamze Gune Eskila. I'm not sure I'm pronouncing your name correctly. Can you hear me? Well, in that case, I'll ask the question on Gamze's behalf again, which is, what do you think about the role of immunogenic cell death in the efficacy of PARP inhibitors? And that's because radiation induces immunogenic cell death in cancer cells.
Well, this is a very exciting arena. Obviously, DDR, DNA repair, is becoming a hot topic in oncology, particularly in the clinical applications. But of course, an even bigger arena is immuno-oncology. And I think there is some really, really exciting interfaces between these two arenas. I believe you might be hearing about some of this a little bit later on from Roger Greenberg. But we do know, for example, that deficiency in DNA mismatch repair can probably, through generation of neoepitopes, help fuel immunogenic responses to those kinds of cancers. And I think there's also the potential there for cancers deficient in other DNA repair pathways. It's also possible, I guess, that PARP inhibitors will further enhance that immunogenicity, if you like, in those circumstances. Although, of course, the ongoing mutation rate in those cells will be relatively slow, hopefully compared to the cytotoxic effect of an acute treatment with a PARP inhibitor. But I think this is a very exciting arena to understand mechanistically, but also to look at this potential for some very exciting combinations for these kinds of agents.
Okay, thank you very much. Next question is from Carlos Ciudad. Carlos, you want to turn your microphone on and tell us where you're from and ask Steve your question? Carlos Ciudad. Okay, well, again, I'll read Carlos's question, and that is, did you also try combinations of PARP inhibitors with CHEK1 or WEE1 inhibitors is there synergy between them?
And again, this is another arena which is really ripe for exploitation and further understanding. On first principles, you might expect there to be some toxicities in these scenarios. I would imagine CHEK1 inhibitors working in the ATR pathway would be selective, would have a combination, would have effects in combination with PARP inhibitors, for example, potentially. But there are so many different combinations and so many different cellular and genetic backgrounds to look at in these scenarios that I think we don't know all the answers. WEE1 inhibitors are also being developed as well, and obviously they are impacting on another aspect of cellular cells' response to DNA damage in the context of cell cycle progression. And I'm sorry I don't have any specific answers to that question other than to say I think these are very exciting and interesting arenas that are being explored now by other groups.
Thank you, Steve. I was able to turn on the microphone. I'm from Barcelona, Spain. Thank you for your answer.
Well, thank you.
Okay, thank you very much. So the next question is from Brahamdut Arya. So Brahamdut, can you turn your microphone on? Brahamdut Arya.
Imagine in a couple of years we're all going to be so fluent at these meetings.
Yeah. So Brahamdut, do you want? I don't know if you can hear me, or if you can you turn your microphone on and ask Steve your question? If not, I'll ask your question on your behalf. Okay, well, I'll ask Brahamdut's question, which is, I found some interesting results using autophagy inhibitors to induce DNA damage in cancer cells as PARP1 inhibitor. Can we use autophagy inhibitors along with first-line drugs to have a combinatorial effect? That is the question from Brahamdut. So autophagy inhibitors in combination.
Okay, well, I can't claim to be an expert in autophagy, but I know more or less of what those pathways do. And I think on first principles, you would expect the potential for combination effects because they are targeting very different aspects of cell function. But there are reports of some interesting interfaces between these autophagy pathways and DNA damage response pathways. And I didn't really dwell on this at the beginning of the talk, but we know that DNA repair and associated DNA damage signaling impact on so many things within the cell, from chromatin structure all the way through to certain metabolic pathways. So I'd be very surprised if there isn't some potential for combining DDR inhibitors such as PARP inhibitors and autophagy inhibitors.
Okay, well, thank you very much. That's all the time we have for questions now. So I'd like to thank Steve very much for sharing his very exciting and important research. And we have a 5-minute break before we start in the next session with Gail Lagoob at 10 to 2. Okay, so we have a 5-minute break now. Thank you very much, Steve.